Alright guys, today I'm going to explain you on lipoprotein metabolism. So as you all know that in lipoprotein metabolism, so the metabolism of chylomicrons, very low density lipoproteins and high density lipoproteins. These are the things we are going to discuss in lipoprotein metabolism. So just to give you a brief introduction to lipoproteins, so we have chylomicrons which are least dense molecules, and then we have VLDL which is very low density lipoprotein, then we have Hdl intermediate density lipoprotein, LDL for low density lipoprotein, and then we have HDL, that is high density lipoprotein. Just to give you a brief idea about apolipoproteins which are important on each of these molecules. For chylomicrons, important apolipoproteins are apolipoprotein B48 and ApoC2 and ApoE. These are the important apolipoproteins. Then the VLDL molecule, very low density lipoprotein, important apolipoproteins on the surface is ApoB100 and then ApoE and ApoC2. For LDL it is ApoB100. For HDL it is apolipoprotein A1. So these are all the important apolipoproteins present over the lipoprotein particles. Now along with this apolipoproteins, HDL particularly, it has got CETP, that is cholesterol ester transfer protein. Then it has got peroxanase, which is an antioxidant enzyme, and then it has got LCAT, that is lecithin cholesterol acyl transferase. With this introduction, let me begin with lipoprotein metabolism. So I'll start with the digestion and absorption of the lipids. So digestion of the absorption of lipid, I have a video on that in the YouTube. So you can go to that video and know the details of digestion and absorption. So I'll be very brief in explaining you the digestion and absorption of lipids here. So the dietary lipid, so the diet has got triacylglycerol, it has got cholesterol ester, it has got phospholipid, some of the free fatty acid, and then it has got vitamin A, D, E, K. All this in the diet. So intestines absorb them. Basically it is absorbed in the brush border epithelium. So after the digestion mediated by pancreatic enzymes. So the pancreatic enzymes, that is pancreatic lipase along with the colipase. I will just write one of the brush border epithelium in the intestine. So the triacylglycerol in the lumen, it is digested into free fatty acids and 2-monoacylglycerol, one molecule of 2-monoacylglycerol done by pancreatic lipase along with the colipase. This needs bile acids and bile salts, emulsification and all that process. So cholesterol ester digested by cholesterol esterase enzyme into cholesterol plus fatty acid. Then the phospholipid is digested into lysophospholipid and fatty acid, and vitamin A, D, E, K. All of that, they all will undergo diffusion, simple diffusion. Only point that you need to remember here is cholesterol absorption is inefficient. Only 55% of the cholesterol is absorbed, 45% just goes in the feces. Once all these molecules are undergoing simple diffusion, they will be re-esterified into triacylglycerol, cholesterol ester, phospholipid and vitamin A, D, E, K. All of them, these molecules will be loaded onto ApoB48, which is synthesized in the intestine. Okay, how the ApoB48 is synthesized? ApoB48 synthesis, it will undergo mRNA editing. So the mRNA for ApoB100 is basically 48% translated. Why? Because in the intestine there is an enzyme called cytidine deaminase. This cytidine deaminase, it is going to convert GAA into UAA. Cytidine deaminase. So GAA, which is coding for glutamine, normally it codes for glutamine. This is converted to UAA. And as you all know, UAA is a stop codon. So this job is done by cytidine deaminase enzyme. I am just writing it as CD here. Cytidine deaminase. Okay. Because of that, so UAA becomes stop codon and translation is stopped. And this happens when 48% of your mRNA for ApoB100 is translated. That's why you get ApoB48 here. So ApoB48, after taking triacylglycerol, cholesterol ester, phospholipid and vitamin A, D, E, K, and this molecule we call it as chylomicron. And this chylomicron is a large molecule, so it cannot be secreted into the blood vessel. So initially it is secreted into lymphatics, then it finds its way into the bloodstream. So once it is there in the bloodstream, your chylomicron, which has got triacylglycerol, cholesterol ester, phospholipid and ADEK. This chylomicron, originally it has got ApoB48, and it's going to, and I'll write chylomicron as CM here. This is going to acquire ApoC2 and ApoE from HDL. So it gets ApoC2, I'll write C2 here, ApoC2 and ApoE. It gets it from HDL. Okay, so once it gets that from HDL, this is a mature chylomicron. So the mature chylomicrons, as they move in the blood vessel, and also note that entire lipoprotein metabolism is going on in the blood vessel. Okay, so as it is moving on in the blood vessels, so I'll write the endothelium here. So consider this is the blood vessel. Let me change the color. This is the blood vessel, and I am just writing the endothelium of the blood vessel. So to the endothelium of blood vessel, LPL is attached, that is lipoprotein lipase is attached to the endothelium of blood vessel with the help of heparan sulfate. Heparan sulfate is a glycosaminoglycan, so it is holding on to lipoprotein lipase. Now your chylomicron is moving in the circulation. As it is moving through this lipoprotein lipase, so the ApoC2, especially this ApoC2 here, it is going to activate lipoprotein lipase enzyme. So in connection with the ApoC2, lipoprotein lipase is activated, and what it does, it is going to break down triacylglycerol present in the chylomicrons and release free fatty acids. And that free fatty acid will be taken up by the peripheral tissue. It gets into the peripheral tissue. Free fatty acid is getting into peripheral tissue, and also it releases glycerol. So basically lipoprotein lipase is going to break down triacylglycerol into free fatty acid and glycerol molecule. So free fatty acid will be used for whatever the needs of the peripheral tissue, for membrane formation and whatever the needs of it. Glycerol will go back to the liver and it will be converted to glycerol 3-phosphate, and glycerol 3-phosphate can be used to make triacylglycerol, or glycerol 3-phosphate can be converted to dihydroxyacetone phosphate, and that can get into glycolysis. That's the fate of glycerol coming from chylomicron, because chylomicrons are formed only when you are in well-fed condition. Chylomicron metabolism is going on in the well-fed condition. And also note that insulin is going to stimulate your lipoprotein lipase enzyme. In the presence of insulin, expression of lipoprotein lipase increases. It means metabolism of chylomicrons also increases. Anyway, with the action of this lipoprotein lipase, so continuous degradation of triacylglycerol occurs. At some point in time, ApoC2 will go back to HDL. There will be release of ApoC2. Okay, so once that happens, you are calling this molecule as chylomicron remnant. So remaining portion of chylomicron, so I'll write it as CM remnant. Chylomicron remnant. This chylomicron remnant, now it has got ApoB48, then it has got ApoE, and also it has got more in, now it is rich in cholesterol, cholesterol ester, then it has got some amount of triacylglycerol. Of course, it has got phospholipid and vitamin A, D, E, K. All these fat soluble vitamins are still there. Okay, now it will be taken up by the liver. So the chylomicron remnants will be taken up by the remnant receptors present over the hepatocyte membrane. So this receptor is ApoE receptor, ApoE receptor, ApoER receptor. Chylomicron remnants will go and bind with the ApoE receptor. Basically, ApoE is recognized by ApoE receptor and it is internalized. Basically, it is going to offload all the content, means cholesterol, cholesterol ester, then vitamin A, D, E, K, some amount of phospholipid, whatever is there in the chylomicron remnant, is offloaded, and also of course the protein is degraded into amino acids. Basically FOB48, FOE, all that is degraded into amino acids. That's the fate of chylomicron remnants. Okay, so overall in this metabolism, what we have seen, chylomicrons are formed in the intestine. They are matured in the circulation by gaining FOC2 and FOE from HDL. Then lipoprotein lipase is activated by FOC2 and it is going to degrade triacylglycerol into free fatty acid and glycerol. Free fatty acid is used by the peripheral tissue wherever that is going on. Glycerol will go back to the liver. Glycerol will be taken back to the liver here. Get into the liver. Glycerol is converted to glycerol 3-phosphate, and this can be going into triacylglycerol formation, or glycerol 3-phosphate converted to DHAP, and DHAP gets into glycolysis. That's the fate of glycerol coming from chylomicrons. Okay, and insulin keeps your lipoprotein lipase active. In the well-fed condition, there is more and more metabolism of chylomicrons. So at the end of it all, chylomicron converted to chylomicron remnant, it has got cholesterol ester, cholesterol, phospholipid, vitamin E, D, K, all that is taken to the liver and offloaded there. This is how your dietary lipids are carried from intestine and they will end up in the liver, except that triacylglycerol portion is used by the peripheral tissue. Each peripheral tissue, they will express different kinds of LPL. So skeletal muscle, the cardiocyte LPL, it has got low Km, it means it's going to act on chylomicrons most of the time, whereas adipose tissue LPL, it has got high Km, it means it's a low affinity enzyme. It will use chylomicron only when there are plenty of chylomicrons in the circulation. Alright, now let me explain you on metabolism of VLDL. So as you all know, under fed conditions, especially when someone takes carbohydrate rich diet, so glucose is converted to acetyl-CoA can be diverted into fatty acid synthesis in the cytoplasm. Okay, so the fatty acids can be synthesized, cholesterol can be synthesized under well-fed conditions, especially influenced by insulin. So whenever there is increase in the synthesis of cholesterol, which is converted to cholesterol ester in the liver by ACAT enzyme, that is acyl-CoA acyl transferase enzyme, and then you make triacylglycerol under fed condition, and then you have vitamin E coming from that vitamin E here, because vitamin E need to be transported out by VLDL, whereas other vitamins, that is fat soluble vitamin A, D and K, they have separate transporters. So vitamin E here and some amount of phospholipid, all this will be loaded onto lipoprotein B100. Okay, so the lipids from smooth endoplasmic reticulum and ApoB100 in the rough endoplasmic reticulum. So it will be fat is transported into the rough endoplasmic reticulum by MTP, microsomal transfer protein. Okay, so that's how you make VLDL here, and this VLDL is secreted into the circulation. Now your VLDL is in the circulation. Write it as VLDL here. This is VLDL, which is called as nascent VLDL, newly synthesized VLDL molecule. So now it has got triacylglycerol, and that's the major composition. Then it has cholesterol ester, phospholipid and vitamin E, and it has B100 on the surface. Now this VLDL, it will acquire, means it will take FOC2 and FOE from HDL, just like your chylomicron. As chylomicron we have seen, it is taking FOE and FOC2 from HDL. In the same way, FOC2 and FOE, it will be given by HDL to VLDL. Now this VLDL is considered as a matured VLDL now. Maturation is going on in the circulation. So the mature VLDL, as it is passing through the LPL, see this is again same process, LPL attached to the endothelium, which was acting on chylomicron before, now it is going to act on VLDL. So LPL is going to degrade triacylglycerol present in the VLDL, and it is going to release free fatty acids taken up by the peripheral tissue, and it is going to release glycerol. Okay, now with the action of LPL, so constantly triacylglycerol is broken down into free fatty acids and glycerol. And also note that VLDL synthesis not only goes on in well-fed condition, it can also go on under fasting condition. Why? Because under fasting condition, there will be lipolysis going on in the adipose tissue. If there is too much of lipolysis, so the fatty acids which are flooding into the liver, those fatty acids can be esterified to make triacylglycerol, and you can synthesize VLDL. And VLDL comes into the circulation. Only thing is, under fasting condition, insulin level is less. Because of this, LPL action can be reduced or decreased. Because of this, metabolism of VLDL can be affected. Otherwise, VLDL can be synthesized under fed condition and also under fasting condition. Anyway, so whether it is in fed condition or in fasting condition, depending on the levels of insulin, LPL is active. So it is going to degrade triacylglycerol into fatty acid and glycerol. So meanwhile, once this happens, at some point in time, there will be release of ApoC2. ApoC2 will go back to HDL. At that time, we call that molecule as ideal. So we got ideal now. Ideal has got, still it has got some amount of triacylglycerol. It has got cholesterol ester. It has got phospholipid, and it has got vitamin E. Now it has got B100 and it has got ApoE, because we have released ApoC2, and that will go back to the HDL. Okay, once it doesn't have ApoC2, so no longer it is going to be catalyzed by lipoprotein lipase. Now what happens to the ideal? Ideal has got three fates. One fate of ideal is it can be taken up by the liver, and that is done by ApoE receptor. ApoE receptor, which are called as remnant receptors. Very similar to what we have seen here for chylomicron remnant. Same remnant receptors can uptake ideal, and all the contents will go to the liver. That's one fate of it. Another fate of ideal is it can pass through the membrane of hepatocyte, that is the sinusoids of the hepatocyte. Over the membrane of hepatocyte, we have an enzyme called hepatic lipase. I'll write that as HL. HL for hepatic lipase. What this hepatic lipase does, it is going to degrade this triacylglycerol. Triacylglycerol is degraded into free fatty acids and glycerol molecule. So with the action of hepatic lipase, so you are degrading triacylglycerol and the molecule is coming out of it. And the molecule that comes out of hepatic lipase will now has very less amount of triacylglycerol, and it has more cholesterol ester, phospholipid, some amount of phospholipid, and it has got vitamin E and less amount of triacylglycerol. Meanwhile, as it is released from hepatic lipase, ApoC2 will go back to HDL. Sorry, not ApoC2. ApoE will go back to HDL. So now your molecule has got only ApoB100 on the surface, and this molecule is called as LDL, that is low density lipoprotein. That's the second fate of ideal. So first fate, what we have seen, it is completely taken up by the liver by ApoE receptor. Second fate is hepatic lipase acting on it, converted to LDL. Third fate of ideal is ideal will undergo a reaction catalyzed by CETP. CETP. The CETP enzyme is cholesterol ester transfer protein. What this cholesterol ester transfer protein does is it is going to transfer cholesterol ester from HDL3 into VLDL, IDL and LDL. Let me write that part for you. So HDL3, it will be something like this. It is rich in cholesterol ester in the center, and it will be having some amount of phospholipid, and on the surface it will have ApoA1. Then it has got three enzymes on the surface, and that is CETP. This is what I am going to explain you now. CETP. Then it has got LCAT, and it has got peroxidase enzyme, and of course it has got all the apolipoproteins except ApoB100 and ApoB48. That's the composition of HDL. HDL has got all the other apolipoproteins except ApoB48 and ApoB100, and additionally it has got CETP, LCAT, peroxidase. So CETP that is there on the HDL, what it does, it's going to interact with VLDL, it's going to interact with IDL, it's going to interact with LDL. All the three enzymes, all the three lipoproteins interact with the HDL molecule, and this is an HDL3 here. HDL3 interacts with VLDL, IDL and LDL via CETP. What this CETP enzyme does? CETP enzyme, it is going to take triacylglycerol from VLDL, is going to take triacylglycerol from LDL, is going to take triacylglycerol from IDL, all three of them. In return, what it does is going to give cholesterol ester to them. To the VLDL, cholesterol ester is given to the LDL, cholesterol ester is given to IDL. So all three molecules will receive cholesterol ester from HDL3. In return, they are going to give triacylglycerol. That is the function of cholesterol ester transport protein. Why this is going on? The importance of this is cholesterol ester that is there in the HDL3. How that comes into the HDL3, I am going to explain that in HDL metabolism. So HDL3 is rich in cholesterol ester. That cholesterol ester is exchanged with VLDL, IDL, LDL for triacylglycerol. Ultimately what happens, what we have seen, VLDL is converted to IDL, IDL is converted to LDL. So LDL is getting rich in cholesterol ester. Okay, it means cholesterol ester that was there in the HDL3 ultimately it ends up in LDL molecule. Why? Because VLDL converted to IDL, IDL is converted to LDL. This LDL, once this LDL is synthesized, matured, this LDL is 40% is taken up by the extrahepatic tissues by LDL receptor. This is an LDL receptor which is going to recognize ApoB100. 40% of the circulating LDL will be taken up by LDL receptor. 60% of it will be taken up by liver. Again by LDL receptor. LDL receptor 60% will be taken up by the liver. 40% is taken up by the extrahepatic tissues. That is how LDL will end up into extrahepatic tissues that are peripheral tissue. So what is there in the LDL? You have cholesterol ester, vitamin E, some amount of phospholipid and very little amount of triacylglycerol, and that's how all your cholesterol ester and cholesterol along with the vitamin E, it ends up into extrahepatic tissues. Extrahepatic tissue, 40% of that, and rest 60% will go to the liver. From the liver, that will come back again in the form of VLDL. So it means redistribution is going on here. So any tissue which doesn't need cholesterol, they are going to pump it out, which I am going to explain you in HDL metabolism. So tissues which needs cholesterol, they express high levels of LDL receptor. That's the concept that you need to remember. Any tissue which needs cholesterol, there will be increased expression of LDL receptor on the membrane. Any tissue which has got plenty of cholesterol, there will be decreased expression of LDL receptor on the membrane. It means tissues which have got plenty of cholesterol, they are not taking LDL inside. Tissues which needs cholesterol, they are going to take LDL inside. In this sense, basically HDL cholesterol, which is taking cholesterol from the peripheral tissue, giving it to VLDL, IDL, LDL, ultimately it ends up in LDL. That will be taken up by 60% of it is taken up by the liver, and again coming back into the circulation by VLDL, converted to LDL. 40% of that is taken up by the any tissues which needs LDL, means which needs cholesterol. In this sense, basically you are doing redistribution. So CETP function is all about redistributing cholesterol from the tissues which has got plenty to the tissue which has got less. Okay, that's how the redistribution is going on for cholesterol molecule. Alright, let me explain now on HDL metabolism. So HDL like synthesis of HDL, it will be from intestine and the liver. So the hepatocytes and the intestinal epithelial cells, they have got an ability to synthesize ApoA1, that is apolipoprotein A1, which is the major apolipoprotein present in HDL, will be synthesized by intestine. So I am going to make ApoA1 here, ApoA1, that is a major apolipoprotein coming from intestine, and it will come from hepatocytes. As it is synthesized and secreted by intestine and the hepatocyte, ApoA1, it brings phospholipid. So it will bring phospholipid along with that. So there will be phospholipid. So ApoA1 along with the phospholipid. Once it is there in the circulation, this ApoA1, it's going to recruit different proteins that are there in the circulation. Basically, it is going to recruit CETP, cholesterol ester transfer protein, which is synthesized by the liver. It's going to recruit peroxidase enzyme, which is an antioxidant enzyme. It's going to recruit LCAT enzyme, lecithin cholesterol acyl transferase enzyme, and it's going to recruit all the apolipoproteins except ApoB100 and ApoB48. Okay, so it has got plenty of proteins on its surface and it has got phospholipid there. The molecule that you see here, which is called as discoid nascent HDL, discoidal because it looks like a disc shape, that's why it is discoidal nascent HDL. This is a discoidal nascent HDL, newly synthesized HDL molecule. Okay, it has got phospholipid there, ApoA1, CETP, peroxidase, LCAT, and also it has got all the other apolipoproteins except B48 and B100. Okay, that is why HDL is acting as a donor of other apolipoproteins to other lipoprotein molecules, like what we have seen before. So I have said chylomicron is gaining ApoC2 and ApoE from HDL. VLDL gaining ApoC2 and ApoE from HDL. Whenever these molecules are releasing ApoC2 and ApoE, they all will come back to HDL. Like I am releasing ApoC2 here. When the chylomicron is converted to chylomicron remnant, ApoC2 is released and that ApoC2 will go back to HDL. When VLDL is converted to IDL, ApoC2 is released and that is going back to HDL. And also, as I said, so IDL, which is converted to LDL, so there will be release of ApoE here, ApoE. This ApoE will be going back to HDL. And also, as it is given here, IDL to LDL, ApoE to go back to HDL. It means HDL will donate apolipoproteins whenever these other lipoproteins they need apolipoprotein molecules. Okay, let's see what happens to nascent discoidal HDL. This nascent discoidal HDL is going to get cholesterol from peripheral tissue. Now here is your peripheral tissue. What we have seen, peripheral tissue takes LDL from circulation by LDL receptor, cholesterol ester along with vitamin E reaches the peripheral tissue. Peripheral tissue uses that cholesterol ester for whatever the needs of that tissue. Just in case if there is plenty of cholesterol in the peripheral tissue, they will decrease expression of LDL receptor on the surface, and also they are going to pump out that extra cholesterol into the circulation. Basically, the extra cholesterol is pumped out by ABCA1 transporter. The name of the transporter which is pumping out extra cholesterol from peripheral tissues to HDL specifically is ABCA1, that is ATP binding cassette transporter A1. This is the type of ATP binding cassette transporter. What it does, so it is going to, when the cholesterol is pumped out, it is in direct, basically it is in connection with the LCAT. The LCAT is here, down here, discoidal nascent HDL. So what I'll do is I'm going to bring that discoidal nascent HDL down here so that I'll be able to explain you better. So what I'm just bringing this discoidal nascent HDL and writing that here. So it has got phospholipid, it has got LCAT here, it has got peroxidase, it has got CETP, and it has got ApoA1. Now ApoA1 is a positive modulator of LCAT. ApoA1 basically activate LCAT enzyme. Okay, when ABCA1 is transporting excess cholesterol out of the peripheral tissue into the blood, and that is taken up by the LCAT enzyme, what this LCAT does with that cholesterol? So the cholesterol here, cholesterol which is coming out, it will be converted into cholesterol ester by LCAT enzyme. Name of the LCAT, basically it is lecithin cholesterol acyl transferase. Okay, what it does, it is going to take cholesterol and it's going to break down lecithin. Lecithin is phosphatidylcholine, PC for phosphatidylcholine. Other name for phosphatidylcholine is lecithin, and it's going to release that lecithin as lysolecithin. Lecithin is converted to lysolecithin. So the difference between lecithin and lysolecithin is lysolecithin do not have a fatty acid. Lecithin is losing fatty acid and becoming lysolecithin. Where did that fatty acid go? That fatty acid is taken up by the alkat and insert that fatty acid into cholesterol at the third carbon, hydroxyl group at the third carbon, and convert that into cholesterol ester. That is the function of alkat. So alkat converts cholesterol into cholesterol ester and it is going to load it into the center of discoidal nascent HDL molecule. So alkat continuously does this. So as the continuous accumulation of cholesterol ester goes on, it is going to be accumulated in the central core of nascent discoidal HDL molecule. That's why it's going to change its shape. Basically, your nascent discoidal HDL molecule will be converted into HDL3 because of the continuous action of alkat enzyme. That is the enzyme which is responsible for conversion of nascent discoidal HDL into HDL3. Because of this, what happens? Your HDL3 has become rich in cholesterol ester, and this cholesterol ester present in the HDL3, now it is ready for exchange done by CETP, which I have already explained you previously. So the CETP which is there on the surface of HDL3 molecule, in fact any HDL molecule, what it does, now it's going to exchange this excess cholesterol ester with the VLDL, IDL, LDL in exchange for triacylglycerol. So with the continuous action of CETP, so your HDL3 is losing cholesterol ester and it is gaining triacylglycerol. Okay, because of this, what happens? Triacylglycerol content increases and cholesterol ester content is still present there. So, and also of course it has got ApoA1 on the surface, it has got alkat, all those enzymes, CETP, peroxidase, all enzymes, all the other apolipoproteins except ApoB100, B48 still there. But only thing is HDL3, now it is converted to HDL2 molecule. This is HDL2. HDL2 has more triacylglycerol, and also it has continued to accumulate cholesterol ester because this alkat here, it continues to work on HDL2 also. Whatever the alkat, that function that we have seen here, alkat continue to do its function. So it means it is continue to load cholesterol ester. On one side, HDL3 is losing cholesterol ester by CETP and gaining triacylglycerol. That's why HDL2 has got more triacylglycerol, but it continues to have cholesterol ester. Although it is losing by CETP, but it continues to contain it. Why? Because alkat is bringing it continuously. So as HDL3 is going into HDL2, alkat continue to work and you still have cholesterol ester here. Okay. Now, what will happen to this HDL2 now? So HDL2 has got two fate. One is HDL2 can go to the liver and interact with SRB1 receptor. And what this SRB1 receptor does? So SRB1 receptor, which is present over the hepatocyte membrane, so it is going to take away cholesterol and cholesterol ester from HDL2 molecule. And also there is an enzyme called HL, that is hepatic lipase, which is besides to hepatic lipase, which is besides to SRB1, which is on this surface of hepatocyte. What it does is going to break down triacylglycerol into free fatty acids and glycerol molecule. So basically HDL2, as it is passing through the hepatocyte membrane, it is going to lose its cholesterol ester and cholesterol, is going to lose its triacylglycerol, and that will be taken up by the liver. So at the end of it all, what happens to your HDL2? HDL2 is coming back as nascent discoidal HDL. It means cholesterol ester and triglycerides are taken by the liver and the rest of the molecule is released, and that's what is nascent discoidal HDL molecule. That's one fate of it. Another fate is HDL2 can interact only with hepatic lipase. HDL2 is interacting only with the hepatic lipase. It's not interacting with the SRB1. It is interacting with the hepatic lipase and losing only triacylglycerol part of it. And the rest of the molecule will come as HDL3 molecule. Okay. So if you degrade only triacylglycerol part of HDL2, retain cholesterol ester. So basically you are bringing it back to HDL3, because as you all know, HDL3 is rich in cholesterol ester. whereas HDL2 is rich in both triacylglycerol and cholesterol ester. So hepatic lipase is taking only triacylglycerol into the liver, rest of the molecule is released, and that is what is HDL3 molecule. So nascent discoidal HDL into HDL3 done by LCAT enzyme, HDL3 into HDL2 is done by CETP and LCAT, both the enzymes. Now HDL2 will be going back to the liver by SRB1 receptor, coming out as discoidal nascent HDL after the action of SRB1 and hepatic lipase. Or if only hepatic lipase acts, it is coming back as HDL3. Now overall what we have done here. So overall we have taken cholesterol present in the peripheral tissue back into the liver. Okay. So the cholesterol, excess cholesterol present in the peripheral tissue is taken up by the HDL, and HDL, nascent discoidal HDL converted to HDL3, HDL3 converted to HDL2, HDL2 has given that cholesterol back into liver. And this journey of cholesterol from peripheral tissues to the liver is called as reverse cholesterol transport. Normally what happens, liver is going to synthesize cholesterol and put it on to VLDL, VLDL is converted to IDL, IDL is converted to LDL, LDL is taken up by the liver, peripheral tissue. That's an onward journey for cholesterol. So cholesterol coming from liver to the peripheral tissue, helped by VLDL, IDL and LDL ultimately. That is an onward journey of cholesterol. Reverse journey, peripheral tissues to discoidal nascent HDL, HDL3, HDL2 to the liver. And that's the reverse journey of cholesterol. And that's what is reverse cholesterol transport mechanism. With this, basically HDL is not allowing cholesterol to accumulate in the peripheral tissue, and that helps in the prevention of atherosclerosis. Thank you.